Hi, my name is Laura Casablanca from National Center for Biotechnology in Madrid, and I'm going to present my paper published in GM entitled "A Signal-Responsive Cooperative Transcription Factor Networks Determines Alveolar Macrophage Identity." Today, I will talk about how macrophages are instructed to specialize in order to perform tissue-specific functions in lung tissue. The lungs are constantly exposed to particles, microorganisms, and potentially harmful agents present in the air we breathe. To protect this organ, the immune system relies on specialized cells called alveolar macrophages, renowned AMs, which act as local defenders in pathological conditions such as infections, inflammation, and cancer, while maintaining proper tissue function. But an important question in immunology is, how do these cells actually acquire their identity and function? In our study, we demonstrated that this process is controlled by a ligand-dependent transcription factor known as the retinoid X receptor, or RXR. We found that RXR acts as a central regulator of gene expression in these cells, and it binds to a specific loci in the chromatin and helps to determine which genes are turned on or off by controlling the transcription of AM's-specific genes. We use a CD11C RXR alpha-beta flock-loss conditional mouse model to specifically delete both RXR isoforms in alveolar macrophages. In the absence of RXR, the cells fail to develop properly. We observe a lower number of alveolar macrophages, defined as CD11B low, a single F5, in accordance with an accumulation of immature AM arrested as an intermediate state of AM differentiation, characterized by a high CD11B marker and the failure to upregulate single F. Importantly, they saw an increase in XM5 and body pie staining, suggesting enhanced apoptosis due to lipid accumulation. This suggests that RXR is not only important for the identity of these cells, but also for their function in their lung tissue. We next investigate the molecular mechanism underlying the phenotype of RXR deficient alveolar macrophages using a multi-omic approach, including RNA-seq, ATAC-seq, and high-throughput segmentation. RNA-seq analysis revealed differences between the three groups. To further characterize the molecular pathways altered in immature AMs, Panther gene ontology termed enrichment analysis, comparing mature cow population with wild-type population, showed that upregulated genes, here in red, were mainly associated with cell activation and immune responses, whereas downregulated genes, in blue, were linked to lipid metabolism pathways. Our findings also show that RXR are required to maintain an open chromatin status, and the deletion of RXR in AMs leads to a global loss of chromatin accessibility, impairing the transcription of genes essential for AM identity and function. Moreover, we generated a wide genome RXR binding profile in AMs using high-throughput segmentation and identified direct RXR target genes, including key transcription factor for AM identity genes, cytokine receptors, and genes involved in lipid and apoptosis. Multi-frame enrichment analysis of RXR binding regions revealed RXR colocalization with key AMs identity transcription factor, including SMAD4, STAT5, downstream regulators of lung environmental signals. To investigate the niche signals responsible for RXR gene induction in AMs, we analyzed cell-to-cell communication in the lung niche through a cell-phone-DB-based analysis. These interactions identify niche signals like GM-CSF, delta ligand 4, or TGFβ, signaling to alveolar macrophages. We found that in differentiating GM-CSF bone marrow progenitors, TGFβ induced RXR alpha expression, whereas delta ligand 4 induced RXR beta, with both signals acting synergistically. Moreover, TGFβ and delta ligand 4 promoted the expression of RXR target genes, including AM-specific genes and the AM core genes, supporting a cooperative role for these niche signals in AM differentiation and identity. Based on our segmentation data, we could demonstrate that RXR and transcriptional regulators acting downstream of these signals share binding patterns near genes involved in AM identity, AM core genes, apoptosis, and lipid metabolism. Homer motif and Retinoid analysis of the resulting peaks confirmed the presence of binding motifs for the added transcription factors in the set, reaffirming the cooperation between them. Moreover, we identified RXRs and PPARγ receptors as the most significant transcriptional regulators. Based on our segmentation analysis for PPARγ, we saw that their co-binding and their consistent heterodimeric transcription regulator support the notion that RXR and PPARγ heterodimer controls many AM core genes, as well as those involved in lipid metabolism, apoptosis, and proliferation. Overall, our findings reveal that RXR plays a key role in a coordinated molecular network to establish and maintain the identity and function of alveolar macrophages. These findings could contribute to the development of new therapies to improve the immune system function in lung tissue. And with that, I would like to thank my lab, the ethnic unit, all collaborators, and funding agencies for their support. Thank you for your attention. If you would like to know more about this work, please feel free to read our paper. Thank you.